Have you ever had one of those moments where you realize a fundamental assumption you've held about something, something you thought you knew inside and out, is just completely wrong?
It's a jarring feeling.
Right. Imagine building decades of screening, of treatment protocols, all on what turns out to be a faulty foundation.
Well, it might sound like a theoretical problem, but in oncology, that's quite literally a matter of life and death. And we're talking about a very specific, highly aggressive subtype of breast cancer. It's called classic diffusely infiltrating lobular carcinoma, or DILC.
Okay, so let's unpack this for you. We are diving into a really fascinating piece of research today. It's an article from the International Journal of Molecular Sciences, and it doesn't just, tweak the definition of DILC, it challenges its entire identity.
It really does.
Our mission in this deep dive is to look at this radical new hypothesis that this cancer, it doesn't start where its name suggests, in the epithelial cells of the breast lobules.
No. The idea is that it comes from an entirely different and, frankly, much more dangerous source, something called epithelial mesenchymal hybrid cells, EMHCs.
And the stakes here, they couldn't be higher, could they?
Not at all. I mean, this is one of those deep dives where the source material is just immense. It's pulling together mammographic imaging, large format histopathology, cell culture analysis.
From data going all the way back to 1977.
All the way back. And despite all those decades of advances, the clinical outcome for this specific subtype, for DILC, it just hasn't measurably improved. And if this research is correct, it means our terminology and the therapies we choose because of it are, well, they're fatally misguided.
So let's start where all the confusion begins, with the name itself, lobular carcinoma.
Right.
That implies it comes from the breast lobules,
Right.
what are they called? The terminal ductal lobular units.
TDLUs, yep. And historically, breast cancers are pretty much mapped to one of three places: the big ducts, the TDLU, or the mesenchyme.
The mesenchyme being the fibrous connective tissue, kind of the framework.
Exactly. And DILC has always been lumped in with the TDLU cancers, but this paper argues that its behavior is just a total outlier.
So how is it an outlier? What's the first clue?
Well, that's the core paradox. The researchers noted that they failed to find any evidence supporting the idea of a lobular epithelial origin. It's just not there. The inconsistencies start the moment you look at the tissue.
Under the microscope.
Under the microscope. And the first huge piece of evidence is this classic finding they call the targetoid pattern.
The targetoid pattern. Okay, that sounds like something from a sci-fi movie. What does it actually look like?
So think of the lobule as a target. The invasive cancer, it surrounds the little acini and the major ducts. It's like an archer hitting the rings around the bullseye.
Okay, I can picture that.
But here's the critical clue. The epithelial cells within those structures, the bullseye itself, they remain totally normal.
Wait, what?
They're pristine, free of atypia, free of LCIS, which is lobular carcinoma, in situ the precancerous stage.
So hold on. If the cancer started from those epithelial cells inside the lobules, wouldn't you expect to see some signs of trouble there? Like the precancerous changes, the LCIS, before it breaks out?
Precisely. You should see a progression from normal to abnormal to invasive. The fact that the invasive DILC is surrounding structures that are completely normal internally, it just strongly suggests the cancer is invading from the outside tissue.
From the framework.
From the framework, not starting inside the structure as the name implies.
Wow. That immediately points the finger at all that surrounding tissue. And the paper mentions that DILC has a ton of that, right?
Yes, an exceptional amount. DILC is defined by an excess of fibrous connective tissue, the mesenchyme. It's a dominant feature. This tumor isn't some tight little ball of cancer cells.
Right.
It's this vast, diffuse process that's embedded in all this fiber and structural material. The sheer dominance of the mesenchyme is basically screaming, hey, look over here.
And this structural difference is exactly why DILC is so difficult to spot on a mammogram.
Absolutely. It behaves so differently. If you look at a cancer that really does start in the lobule, let's call it an acinar adenocarcinoma of the breast, or AAB, it grows outward. It pushes. It creates this convex contour against the fat tissue.
So it's like a distinct pushing mass that you can see. It stands out.
It stands out. But DILC characteristically has concave contours. It pulls the tissue in. It infiltrates, weaving itself into the architecture instead of pushing it aside.
And when a tumor has a concave contour, it's basically hiding.
It's exceptionally good at hiding in normal, dense tissue. It's incredibly hard to tell it apart from just benign scarring or architectural distortion.
That's a massive clinical problem. If a mass is pushing out, you see the boundary. If it's pulling tissue in and just melting into the background, It's a ghost.
It is, especially when you add in the fact that it has a critical absence of malignant-type calcifications.
Which are often the first sign of other cancers.
The first, easiest to spot sign. So DILC often stays hidden until it's big enough to be felt, or until it's caused this massive late-stage distortion. The clinical implications are devastating.
Okay, so the pathology is wrong. The imaging looks wrong for an epithelial cancer. The tissue itself is dominated by mesenchyme. The origin has to be in that framework tissue.
That's the only logical conclusion, and all of this structural and visual evidence acts as this perfect bridge to the molecular hypothesis. The only component left that explains all these features is the mesenchyme.
And that's where they bring in the hybrid cells.
Yes. The paper posits that DILC arises from epithelial mesenchymal hybrid cells, EMHCs. And this is a game changer because it means the cell has characteristics of both structural tissue and invasive migratory tissue.
Let's pause on that because that hybrid status, that sounds terrifying. In cancer, epithelial cells are the builders, right? They form structures.
Correct. And mesenchymal cells are kind of like the repair crew. They move, they invade, they heal wounds.
But a hybrid is...
It's the worst of both worlds. An EMHC can proliferate and grow like an epithelial cell, but it can also detach, migrate, and weave through tissue like a mesenchymal cell. This hybrid state is almost always associated with the most aggressive, adaptive, and drug-resistant cancers. They have maximum flexibility.
Which makes perfect sense for a cancer that presents with that diffuse tissue-pulling structure. And they didn't just guess this, they proved it. They actually cultured cells from DILC tumors.
They did, and the results from the cell culture were just incredibly compelling. First, the cells showed remarkable persistence. I mean, they survived for over a year in culture,
Wow.
reaching the 15th passage. That alone tells you these are resilient stem cell-like cells, but they're slow-growing, and their morphology was described as fibroblastoid and mesenchymal stem cell-like. Definitely not a sheet of epithelial cells.
And what about their ability to form tumors?
They didn't need any coaxing at all. The cells formed these microtumors spontaneously, right in a simple culture dish. No special scaffolding, no extracellular matrix needed.
So they are programmed to be cancerous.
Absolutely. And what's really fascinating is that the pattern of those microtumors in the dish was reportedly similar to the tumor's appearance on the mammogram. They literally replicate their disruptive signature in vitro.
Okay, but this brings up a crucial point for anyone listening. The culture showed low proliferation, and the tumors have low Ki-67 scores, like 5 or 10%.
Right, Ki-67 being the standard marker for how fast cells are dividing.
So if this tumor is so slow growing, why is DILC so deadly? Doesn't speed usually equal fatality in cancer?
That is a fantastic, critical question. The reason is the hybrid nature of the cell. They might not divide rapidly, but their mesenchymal side makes them incredibly effective at invasion, diffusion, and spreading to distant sites. The DILC cell is slow, but it's tenacious, it's adaptive, and it's highly mobile.
So it gets the job done by stealth, not speed.
Exactly. And that low Ki-67 is what often tricks people into thinking these tumors are low and aggressive on paper. But in reality, their hybrid nature makes them far more dangerous.
So the pathology and the behavior all line up. What was the molecular smoking gun? The evidence that finally proved these cells were truly hybrid.
It was a beautiful molecular fingerprint. First, they looked for mesenchymal stem cell markers. The cells were positive for the key ones, CD73, CD90, and CD105.
Okay.
And just as importantly, they were negative for CD34, which helps rule out some other cell lines. That combination establishes a clear stem cell identity in the mesenchymal family.
So that's the mesenchymal side of the hybrid confirmed.
Exactly. Then, for the hybrid part, they showed a dual phenotype. They were positive for epithelial markers like cytokeratin, AE1, AE3.
The proteins that literally define epithelial cells.
Right. And at the same time, they were positive for mesenchymal markers like alpha-smooth muscle actin or alpha-MNA.
Which is all about movement and contractility.
Yes. So this simultaneous expression of markers from two totally different lineages, that confirms their hybrid nature.
They're biologically conflicted, and that conflict is their superpower. And they had other markers that signal a major threat too, didn't they?
They did. They expressed pluripotency markers, things like OKT4 and NANOG. These are usually found in embryonic stem cells, and they signal incredible plasticity, the ability to adapt and change. Their presence just reinforces this superior tumorigenic potential.
This molecular fingerprint, it changes everything. And this is where we shift from the lab to the clinic.
Mm-hmm.
What does it mean for treatment that we've been calling these hybrid malignancies purely epithelial?
It means profound therapy resistance. If your guiding belief is that DILC comes from epithelial cells in the lobules, you select treatments targeted for epithelial cells, endocrine therapy.
All standard
The
playbook.
standard playbook. But the problem is EMHCs are inherently programmed for survival and adaptation. They are known to have a poor prognosis and, crucially, resistance to therapy targeted for malignancies of breast epithelial origin.
We're targeting the wrong molecular engine. And that finally explains why, with all our medical progress, the outcomes haven't improved for this specific cancer.
Absolutely. Think about the impact. DILC is maybe 5 to 8% of all breast cancers, but because it's so deadly and resistant, it accounts for a disproportionately massive percentage of all breast cancer deaths. We've been losing this specific fight because we've misidentified the enemy.
The source material provides just a stunning comparison that makes this undeniable. Let's talk about case 3, the patient who had this, natural experiment going on in her own body.
Oh, case 3 is incredible. This was a patient with 2 simultaneous but different tumors, one in each breast.
Okay.
In her left breast, she had a massive 75 millimeter DILC. The histology showed all the classic features: diffuse infiltration, mesenchymal dominance, and significantly the absence of E-cadherin.
And E-cadherin is the cellular glue, right? It keeps epithelial cells stuck together. Losing it means they're ready to move.
Correct. The loss of E-cadherin is a hallmark of the shift towards being mesenchymal and invasive. It confirms DILC's nature.
Okay, so that's the left breast. What was in the right?
In the right breast, there was a much smaller 18 millimeter acinar adenocarcinoma of the breast, an AAB. And remember, AAB is our gold standard for a true lobular cancer, one that really does start in the TDLU's epithelial cells.
And it's E-cadherin?
Its histology showed strong positive E-cadherin staining.
Wow. So in one person, at the same time, you have a side-by-side comparison. The cancer we call lobular, DILC, which has lost its epithelial glue, next to a cancer that is truly of lobular epithelial origin, the AAB, which has kept its glue.
It's the smoking gun. It just reinforces that DILC and AAB are fundamentally different diseases. They have different molecular behaviors, and they absolutely require different terminology and different treatment.
They shouldn't even share the same name.
They shouldn't, because they don't share the same origin story.
And that's the paper's powerful conclusion. It says that as long as we call this lobular carcinoma, real progress in controlling it is just. It's unlikely. Correcting the name is step 1.
It is. A new terminology, one that specifically points to its actual mesenchymal or hybrid origin, is the absolute necessary first step. That's how you start developing new, appropriately targeted therapies that actually go after the EMHC.
This research just proves the immense power of correct identification. This isn't just about updating some guideline. It's about changing decades of protocols because we finally know who the real enemy is.
That's the core paradigm shift here. DILC is very likely a malignancy of mesenchymal, of hybrid cell origin, not epithelial. And this finding is supported by this powerful multimodal stack of evidence: the imaging, the histopathology, and the cell culture markers that confirm it.
Improving this epithelial mesenchymal hybrid origin, it just opens the door for developing therapies that can finally get better control of this really complex, highly fatal subtype. It forces us to rethink everything.
It does. And the next immediate question for researchers, and maybe something for you to think about, is this. Since these hybrid cells show such strong resistance to our traditional therapies, what new pathways can we target? We know they express those key pluripotency markers like NANOG and OKT4. That's a vulnerability. That's a switch we might be able to find a way to flip off.
It really makes you consider how quickly a single change in scientific understanding, just correcting the origin story from epithelial duct to hybrid mesenchyme, can cascade into a complete overhaul of clinical terminology and, most importantly, save the lives of people who are currently fighting a ghost.